Welcome to part 5 of this 7-part video series on troubleshooting stuck malolactic fermentations. In this video, we will be focusing on the role of medium chain free fatty acids in MLF. Join us as enologist and senior application scientist Duncan Ham from Hansen explains the impact of these compounds on the fermentation and shares tips on how to address the challenges they may present.
Hello, and thank you for tuning into our short video on stuck malolactic fermentation from medium chain free fatty acids. So what is the problem around medium chain free fatty acids? Well, this family of compounds occurs in some wines. There's 3 particular compounds within the family which are most toxic, represented here as C8, C10 and C12, which is octanoic, decanoic and dodecanoic acid respectively. Now, unfortunately, they are toxic to wine bacteria, so if their concentrations in a given wine are high enough, they can affect the population that would otherwise do MLF, resulting in a stuck malolactic fermentation. So what are the causes of these medium chain free fatty acids? Well, they're very much derived from yeast, from the species Saccharomyces cerevisiae. Whether they're produced or not does depend on 2 things. First of all, the yeast strain used and the winemaking conditions. And that's because they're generally produced as a response to stress. So in a wine being fermented with a very high concentration of sugar, the osmotic stress can lead to fatty acids. Likewise, a juice being fermented with a very, very low pH can also lead to them. So how are they diagnosed? They're not an easy thing to measure. Generally, you need to use HPLC, so high performance liquid chromatography, to measure concentrations of the fatty acids. But just knowing the yeast strain employed during the primary fermentation can be enough to point to their presence. They're also extremely easy to treat. Now, we would strongly recommend using Bactivate, and that's because Bactivate is able to bind medium chain fatty acids, thereby making them unavailable to the bacteria and allowing MLF to continue. To help demonstrate the importance of controlling medium chain free fatty acids, we have some experimental data that we've run here on the screen. We've taken a Chardonnay with 13% alcohol at pH 3.4, and we've inoculated with a strain of Unococcus, one of our viniflora strains. And we've done a few treatments where we've also inoculated with decanoic acid, C10. Now, the purple line on the left and right graphs, so on the left, the malic acid, and on the right, the cell count, it proceeds normally. Malolactic fermentation completes in 7 days, and there's a healthy population. But as we go through the blue at 5 parts per million, the red one at 10 parts per million, and the green one at 15 parts per million, as we increase the level of decanoic acid, we do see increased inhibition. So we see a slower malolactic fermentation, and actually it's a 15 parts per million. We see pretty immediate cell death. So this highlights it really is important to control these fatty acids. Thank you very much for listening. I do hope that was constructive.